Welcome to Touching Life Deeply. This research series is published on the channel at 8:00 p.m.
Thank you for tuning in, dear listeners.
Today, we're embarking on really a deep dive into the very core of what makes us tick. We're exploring the molecular instruction manual for life itself, DNA.
Yes, we've gathered quite a bit of material looking into the fundamental principles of human genetics and molecular biology. Our aim here is really to unpack how DNA stores information, how it gets copied, and how that information is used to build and maintain, well, everything alive.
Think of it as trying to understand the master blueprint, this incredibly detailed plan written in a molecular code. It governed, everything from eye color to how our cells function day to day. We could break down the essentials.
Absolutely, focusing on the key mechanisms our sources highlight.
So let's start right at the beginning, the blueprint itself. What is DNA, fundamentally?
Okay, yeah. So the sources consistently describe it as that iconic double helix. You picture two strands twisted around each other, right? And the backbone of each strand is made of sugar deoxyribose and phosphate groups. They're linked by these phosphodiester bonds, which gives the strand a direction. We talk about 5' to 3'.
Okay, and the actual information that's in the rungs connecting the strands,
Yeah,
the
it's
basis.
adenine, thymine, guanine, and cytosine, ATGC. That's the code.
And the really crucial part, something that comes up again and again in the research, is how they pair up, isn't it?
Absolutely critical. A always pairs with T, and G always pairs with C. This specific pairing, this complementarity, it's not just structural. It's the absolute key to holding the information and, crucially, how it gets copied accurately.
Which brings us to the next point. A blueprint isn't much use if you can't make copies. DNA replication.
Precisely. Before a cell divides, it has to duplicate its entire genome, and with incredible accuracy. The process is called semi-conservative.
Semi-conservative, meaning each new DNA molecule gets one of the original strands and one newly made strand.
That's it. The original acts as the template, and enzymes do the heavy lifting. First, you have enzymes like helicase that unwind and separate the two strands of the double helix.
Okay, so it unzips it.
Right. Then, the main players, DNA polymerase enzymes, step in. They read the template strand and add the correct matching bases. A opposite T, G opposite C, building the new complementary strand.
But there's a catch, isn't there? With the directionality.
Yeah.
The 5' to 3' thing.
Ah, yes. DNA polymerase can only build in one direction, 5' to 3'. And since the original strands run in opposite directions, they're anti-parallel. This creates, well, a bit of a puzzle for the cell.
So how does it solve that?
Well, on one template strand, called the leading strand, synthesis is pretty straightforward. It's continuous, following the unwinding helix.
Okay, smooth sailing there.
But the other strand, the lagging strand, it's oriented the wrong way for continuous synthesis. So the polymerase has to work backwards, away from the replication fork, in short segments.
Short segment.
Yeah, these are called Okazaki fragments. It synthesizes a little piece, then jumps back to make another as more DNA unwinds. Then another enzyme, DNA ligase, comes along and basically stitches these fragments together into a continuous strand.
Wow, okay, so quite an intricate process just to copy one strand backwards.
It really is a beautiful solution. And another important point our sources mention is that this whole copying process doesn't just start at one end of a huge chromosome.
No, that would take forever.
Exactly. Replication actually kicks off at multiple specific locations along the DNA called origins of replication. This creates multiple replication bubbles that grow and merge, allowing the entire genome to be copied much more quickly.
Makes sense. Efficiency is key.
Oh.
Now, what about the very ends of the chromosomes? I remember reading something about telomeres being important there.
Telomeres.
Yeah.
Yes, very important. There are these repetitive sequences of DNA right at the tips of our chromosomes. In humans, it's typically GGGTTA repeated many times.
Like protective caps.
Sort of like the plastic tips on shoelaces, yeah. They protect the important coding regions of the DNA near the ends. The issue is the standard replication machinery can't fully copy the very, very end of that lagging strand.
So with each replication, a little bit gets lost.
In most of our body cells, yes. The telomeres get progressively shorter with each cell division, and our sources really emphasize this link between telomere shortening and cellular aging, or senescence.
So eventually they get too short, and the cell stops dividing.
Often, yes. It's like a molecular clock. Though there are conditions mentioned, like dyskeratosis congenita, where problems with telomere maintenance seem to cause issues earlier.
Is there any way to counteract this shortening?
There is an enzyme called telomerase. It can add those repeats back onto the ends. But importantly, it's usually only highly active in certain cells, like stem cells, and unfortunately, often in cancer cells too. Not so much in regular adult cells.
Interesting. So managing telomeres is a big deal for cell lifespan.
Definitely. Now, even with all this careful machinery, copying billions of DNA bases isn't always perfect, and DNA itself can get damaged.
Right. Errors can slip through during replication, mutations, and things like UV light or chemicals can damage the DNA structure too.
Absolutely. But cells aren't helpless. They have really sophisticated repair systems. It's critical to maintain the integrity of the genetic blueprint.
What kind of repair systems are we talking about?
There are several pathways. One example highlighted is mismatch repair. This system catches errors where the wrong base was inserted during replication, like an A paired with a G accidentally.
How does it know which base is wrong?
It often has ways to figure out which strand is the newly made one, maybe by looking for chemical tags, and it corrects the base on that new strand, assuming the older template is correct. It's quite clever.
And what about more serious damage, like breaks in the DNA?
Yeah. For really damaging things like double-strand breaks, there are other pathways. Some are designed for high accuracy if a template is available, but others are more like, well, emergency repair crews. They're sometimes called error-prone.
Airpro. That sounds risky.
It can be. The priority is just to patch the DNA back together to prevent the chromosome from falling apart, which would likely kill the cell. So it might fix the break, but introduce a small mutation in the process. It's a trade-off. Survival sometimes comes at the cost of perfect fidelity.
Unnecessary risk, perhaps. Okay, so we have the DNA blueprint, ways to copy it, ways to fix it. How does the cell actually use the information stored in the genes?
Right. This gets us to what's often called the central dogma of molecular biology. The general flow of information is DNA to RNA to protein. Proteins are the molecules that do most of the work in the cell.
So the first step is getting the information from DNA into RNA.
Exactly. That process is called transcription. It's like making a temporary working copy of a specific gene's instructions. Instead of copying the whole book, you just copy one recipe you need right now. This happens inside the nucleus in our cells.
And there's another polymerase enzyme involved?
Yes, RNA polymerase. For genes that code for proteins, it's mainly RNA polymerase II. It reads the DNA template strand of a gene.
But it makes an RNA copy, not DNA.
Correct. And RNA uses uracil U instead of stymine T. So where the DNA has an A, the RNA gets a U. Otherwise, the base pairing rules apply G pairs with C.
Is it just randomly copying genes? How does it know where to start and stop?
No, it's highly regulated. Our sources emphasize this. RNA polymerase binds to specific DNA sequences called promoters, which are located near the beginning of a gene. Often there's a key sequence element like the TATA box within the promoter.
Okay, so the promoter is like the start here sign.
Pretty much. But there's more. Other DNA sequences called enhancers can also have a big impact. These can be quite far away from the gene itself, but they can loop around and interact with the promoter region to either boost or dampen transcription.
So like volume controls.
That's a good analogy. It allows for really fine-tuned control over how much of a particular RNA message gets made. This regulation is absolutely fundamental to how cells specialize and respond to their environment.
Okay. So the RNA copy, the messenger RNA or mRNA, is made. Is it ready to be used right away?
Not quite, especially in eukaryotes like us. While it's still in the nucleus, the mRNA undergoes some crucial processing steps.
Like what?
First, a special cap, a modified guanine nucleotide, is added to the 5' end. And a long tail of adenine bases, the poly A tail, is added to the 3' end.
Why add those?
They're really important. They help protect the mRNA from being degraded, they help signal that it's ready to be exported from the nucleus, and they play a role in starting protein synthesis later on.
Okay, protection and export signals. What else happens?
The other major thing is splicing. It turns out that most of our genes are interrupted. They have coding sections called exons, separated by non-coding sections called introns.
Introns. So parts that don't actually code for the protein.
Exactly. Splicing is the process that removes these introns and joins the exons together, creating a continuous coding sequence in the mRNA. It has to be incredibly precise.
Wow, like editing out the unnecessary bits.
Precisely. And here's something really fascinating our sources highlight. Alternative splicing.
Alternative splicing.
Yes. For many genes, the splicing machinery can choose to include or exclude certain exons. So from the same initial RNA transcript, the cell can actually produce different versions of the mRNA by combining the exons in different ways.
Right, so yes, multiple protein isoforms, they're called. They might have slightly different functions or be used in different types of cells. It dramatically increases the coding potential of our genome. It's a major source of protein diversity.
That's incredible efficiency, using the same gene in multiple ways. It really is. So once the mRNA is capped, tailed, and spliced, it's considered mature and gets exported out of the nucleus into the cytoplasm.
And that's where the next stage happens, building the protein.
Yes, that's translation. This is where the sequence of codons, those 3 base units in the mRNA, is read and used as instructions to assemble a chain of amino acids.
And this happens on ribosomes.
Exactly. Ribosomes are complex molecular machines made of ribosomal RNA, rRNA, and proteins. They are the protein synthesis factories. Interestingly, the rRNA component itself actually catalyzes the key step forming the peptide bonds between amino acids.
So the ribosome reads the mRNA. How does it know which amino acid corresponds to which codon?
That's the genetic code. It's like a dictionary where each 3-letter codon specifies a particular amino acid. There's a specific start codon, AUG, which signals where to begin translation and also codes for the amino acid methionine.
Okay, start signal. And how do the amino acids actually get brought to the ribosome?
That's the job of transfer RNA, or tRNA molecules. Each tRNA molecule is specific for one type of amino acid, and it carries that amino acid to the ribosome.
Has it know where to go?
The tRNA also has a crucial 3-base sequence called anticodon. This anticodon matches up. It's complementary to the codon on the mRNA that specifies its particular amino acid. So the tRNA docks onto the ribosome at the right spot.
A molecular delivery service, matching the code.
You could think of it that way. As the ribosome moves along the mRNA, codon by codon, the correct tRNAs bring in their amino acids. The ribosome helps form a peptide bond between the incoming amino acid and the growing chain, and the process repeats. Until the ribosome hits one of 3 stop codons, UAA, UGA, or UAG. These don't code for an amino acid. Instead, they signal the end. Special proteins called release factors bind, and the finished polypeptide chain is released from the ribosome.
This can happen quite fast, right?
Oh, yes. A single mRNA molecule can actually be translated by multiple ribosomes at the same time. Imagine a string of ribosomes all moving along the same mRNA, each churning out a copy of the protein. This structure is called a polyribosome or polysome. Very efficient.
Okay, so now we have this chain of amino acids, the polypeptide. Is that the final working protein?
Not yet. That linear chain is just the primary structure. To become functional, it has to fold up into a very specific, complex, 3-dimensional shape. The shape determines the protein's function.
And does that just happen automatically?
Sometimes for smaller proteins, but often, especially for larger, more complex ones, the folding process needs help. Our sources mention chaperone proteins.
Chaperones, like chaperones at a dance.
Kind of. They help guide the folding process and prevent mistakes, like proteins sticking together improperly before they're fully folded. Some chaperones, like the HSP60 type, even create these little isolated chambers where a single polypeptide can fold correctly without interference from the crowded cellular environment.
That's amazing. What happens if a protein doesn't fold correctly?
That's called misfolding, or denaturation. Misfolded proteins usually lose their function. Worse, sometimes they can clump together, aggregate, and become toxic to the cell.
And this relates to disease, doesn't it?
Absolutely. The sources explicitly link protein misfolding to several significant diseases. Alzheimer's disease involves aggregation of misfolded proteins in the brain. Cystic fibrosis is another example.
What happens in cystic fibrosis?
A very common mutation causes the deletion of just one amino acid phenylalanine at position 508 in a protein called CFTR. This tiny change causes the CFTR protein to misfold slightly. The cell recognizes it as faulty and usually destroys it, so it never reaches the cell membrane where it's needed to transport chloride ions. That leads to the symptoms of the disease.
Wow. One amino acid missing, and the whole protein fails because it can't fold right. It really underscores the importance of that final shape.
It really does. Every step from DNA sequence to folded protein has to be right.
We've touched on regulation here and there, but let's circle back to that. The cell isn't just making all proteins all the time. How does it control which genes are turned on or off?
Yeah, gene regulation is absolutely fundamental. Different cell types need different sets of proteins, and even the same cell needs different proteins at different times. So controlling gene expression, which genes are transcribed and translated, is critical.
What are some ways the cell does this?
Well, one major mechanism discussed in our sources involves chemical modifications directly to the DNA itself. specifically methylation.
Methylation, adding methyl groups.
Exactly. Usually adding a methyl group, CH3, to cytosine bases. Often where a C is followed by a G, these are called CpG sites.
And what does adding this methyl group do?
It generally acts to silence or repress gene activity. Heavily methylated regions of DNA tend to be transcriptionally inactive. It's like putting a do not read sticker on that part of the blueprint. And importantly, these methylation patterns can be copied when the DNA replicates, so the silenced state can be inherited by daughter cells.
So it's a form of cellular memory.
In a way, yes. Classic examples are X chromosome inactivation in female mammals. One of the two X chromosomes in each cell is largely silenced by methylation. Another is genomic imprinting, where for certain genes, only the copy inherited from the mother or the father is active due to methylation patterns established in the egg or sperm.
Fascinating. It adds another layer of control beyond just the DNA sequence itself.
Definitely. And it reminds us that even the parts of the DNA that don't code for proteins, which is about 98% of our genome, are incredibly important.
The so-called junk DNA.
Right, which we now know isn't junk at all. Much of it contains these regulatory sequences like promoters and enhancers we talked about, sequences involved in DNA packaging, structural elements like telomeres. It's all part of the complex system.
So pulling all these threads together, why is understanding this molecular world, replication, transcription, translation, regulation, why is it so important for us, for the listener?
Because, well, these are the absolute fundamental processes of life. They are happening in virtually every cell in your body right now.
And variations or mistakes in these processes, they have consequences.
Exactly. Mutations in the DNA sequence can alter how a gene is transcribed, how the mRNA is processed, how the protein is built, or how it folds. This can lead to a protein that doesn't work correctly or isn't made at all.
Which can manifest as genetic disorders, or contribute to complex diseases like cancer, or even influence aging, like the telomere connection we discussed.
Understanding this basic molecular biology gives us really the foundational knowledge to grasp health and disease at the deepest level. It helps explain why certain conditions occur and points towards potential ways to intervene.
As we wrap up this deep dive, it's worth pausing just to think about the incredible coordination involved.
The sheer scale and precision, billions of base pairs copied, genes selectively read, intricate mRNA processing, ribosomes assembling proteins amino acid by amino acid, chaperones ensuring correct folding.
All happening simultaneously in trillions of cells, and how one tiny alteration in that initial DNA blueprint can cascade through all these steps to have potentially profound effects on an entire organism.
It's truly mind-boggling, thinking about this intricate molecular dance, this perfectly tuned system. What does its complexity suggest about the very nature of life and biological systems? It's a question that keeps scientists exploring.
And finally, some of the information in this deep dive has been compiled from public sources and scientific publications. We are not responsible for any misuse of the content beyond its intended purpose of education and raising awareness. Don't forget to follow Touching Life Deeply so you won't miss our next meaningful insights.